Today, our presenter is Dr. Daniel Ohlsen. Daniel is a botanist at the Royal Botanic Gardens, Victoria, and he specialises in fern taxonomy and systematics. He completed his PhD research at Melbourne University, where he was focusing on the fern family Asplanaceae. And currently, Daniel's role at the Gardens involves contributing to Vic Flora and Flora of Australia, taxonomic and phylogenomic research on ferns, teaching, surveying, and more recently, coordinating the new spore bank. And it's the latter subject that Daniel's going to talk about today, his recent work on the fern spore bank. So I'll hand over directly to Daniel. Thank you very much, Daniel.
Thanks, Chris. So today I'm coming from Frankston, Kananook on Bunurong country. I'd just like to recognise those people, past and present and upcoming, and recognise that sovereignty was never ceded. I'll share my presentation slides. So that should be up now, is that right? Yep, great. So what I'll do today is I'll talk about how RBGV has gone about building a spore bank, and also talk about how far we've got along with that in terms of our progress to date, and just give you a bit of background into what we're dealing with ferns and how that is best done. So here's some species of ferns that we've currently sampled for the spore bank so far. These are all threatened species from Victoria: Splenium apendiculatum, Davallia pixidata, and Pneumatoderus penidra. That's my email on the screen too, so if you've got any questions at any time, you can just email me. I'm happy to answer any questions on this or any other things you might want to ask me. So before I go into any further detail of the spore bank, I'll just have a bit of a discussion about what plants we're dealing with here. So ferns reproduce by spores produced on the undersides of leaves or on their margins. So if I just have a point to this photo here, this is a typical fern that shows that the spores are produced on the undersides of the leaves, which in ferns are called fronds. So those black spots on the undersides, they're actually aggregations of capsules that contain the spores. So there's lots and lots of spores produced on those undersides there. In this fern species here, the spores are produced on the margins of the leaves. So these little brown bits right on the edges of the leaves there are parts of the leaves that become folded over to protect those aggregation of spore capsules. Another group that we could look at too that also produce spores instead of seeds are the lycophytes. So an example of a lycophyte is shown here. This is Phlegmaurus varius. It's a Victorian species, and they're commonly called club mosses, selaginella, or isoetes. And in the past, they've often been studied alongside ferns because they've been referred to as fern allies, simply because they're vascular plants, but they reproduce solely by spores rather than by seeds like pines, conifers, and flowering plants. But now that we've got a better understanding of how different plants are related to one another, we've now realized that lycophytes are a separate lineage completely to ferns. And in reality, the fern allies are actually the seed plants. Seed plants are more closely related to ferns than lycophytes. So I tend to refer to ferns and lycophytes separately, just based on that understanding of their relationship to one another. So lycophytes differ dramatically in their morphology to ferns. They reproduce by spores, but this time the spores are produced in cones, usually near the ends of stem, so at their apices. So in this species here, there'll be little clusters of yellow spore capsules at the end in there. In some species of lycophytes, they're actually aquatic and they produce dilated leaf bases that contain those spore capsules. So the genus Isoetes is like that. There are 123 native fern and lycophyte species in Victoria. That's around about a quarter of Australia's native fern and lycophyte species. So it's relatively depauperate in terms of species richness compared to the tropics. The tropics in Australia are far richer. But despite the fact that we don't have all that many species compared to those other areas, they're still an important component of our vegetation. In some areas, particularly moist gullies, they're one of the more conspicuous components of that vegetation. So you think about tree fern gullies, ferns are the majority of the biomass in a lot of cases. So they do contribute quite a conspicuous and important component of the vegetation in Victoria. They're 4% of Victoria's vascular plant species, so not nearly as rich as flowering plants and conifers. Well, actually, they're more diverse than conifers, but the other vascular plants are far more species rich. Isoetes pusilla is our only endemic species, so the only species that's entirely restricted to Victoria. And compared to flowering plants, which are 8% endemic, much lower than those plants. And that, if you think about how ferns reproduce by tiny spores, that sort of makes sense because spores often disperse long distances in air currents. So the chances are that they haven't been quite as contained as what flowering plants are that reproduce by seed. So they're much larger and have a lower propensity for long distance dispersal. 39 Victorian fern and lycophyte species are rare and threatened. So a reasonable proportion of our fern flora is threatened. The main goal of the spore bank is ex situ conservation of fern and lycophytes. And how we're going to do that is by collecting spores and keeping them in long-term storage. And from that, we could use those spores for potential future reintroductions. So if a fern species was to go extinct in a particular area, those sources of spores could become useful for reintroducing into those areas, particularly if the spores were from a similar or a nearby or the same location. And they can also produce cultivated collections as well. So augment whatever collections we currently have in the botanic gardens and also produce captive populations of those species. Spores are absolutely minute. So mostly they're around about 25 to 55 micrometers long. And just to give you an idea of what size that is, I've got a photo in the lower left there that shows all those dots, which are Blechnum wattsii spores. And next to those spores is a ruler for measurement there, just to give you a bit of perspective. Each one of those intervals is a 1 millimeter interval. So as you can see, there's lots and lots of spores that can fit within 1 millimeter. So 1 millimeter square could potentially have 100, if not 1,000 spores in it. So that has a big implication on how effective a spore bank can be for ex situ conservation. If a large number of spores can be stored in such a small space, it means that we can have a quite good representation of the total genetic diversity of a species all contained within storage. So in one collection of one population, we can collect from multiple plants and have thousands, if not millions of spores stored all within a small area or small volume. And we can also collect multiple populations from across the state as well. and still be able to fit it all within a reasonably small volume. So that's potentially a really good advantage to having spore banks, particularly compared to seed banks, where seeds might be a little bit larger and the ability to store so much of that genetic diversity isn't quite there with seeds that are a bit bigger and a bit more space consuming. Most ferns occur in moist vegetation types and are particularly vulnerable to increase in fire and drought. So this project of starting a spore bank couldn't come at a more crucial time, especially with some of the more devastating bushfires we've had recently that can have a huge impact on these plants. So they're among one of the more vulnerable groups of Victorian plants because of all that increase in fire and drought that we might expect to see in the coming future. But despite this, spore banks have lagged behind seed storage. So if I have a look at some of the statistics worldwide at the moment, around about 1,500 seed banks exist, but there are less than 10 fern spore banks in existence worldwide that have a reasonable number of fern species protected within them. And in that 10, there's even less that actually have those spores preserved in conditions that are optimal for long-term storage of spores. They're more of a temporary measure. So in Victoria, in the past, we've had, and we still currently do, a conservation seed bank that's been running for 17 years. So it's only now that we're starting to do look at storing spores as well. I've got a picture here on the bottom right that's of a place called Bryce Gorge. It kind of demonstrates where a lot of fern species would be expected to occur in Victoria. They're often around gullies and creeks in wet places. And this particular gorge is a hotspot for fern diversity. It has several species that are only found in and around that gorge, likely because that gorge has acted as a refugium, so a protected chasm against fire and extremes of temperature and water deficiency. So bit of a demonstration that change in water content can have a huge, profound effect on distribution of ferns. So how do you go about starting up a spore bank? Well, first you need to collect the material. So the material that we're collecting is all from natural populations, and we want to collect throughout their ranges in order to try to collect as much of the genetic diversity that might be underlying in those species. So I'm aiming initially to collect maybe 10 to 20 populations across the range for some of the widespread and common species. But for the rarer species, we'll be focusing on trying to get most of those populations that are currently in existence collected and represented in the spore bank. Also use some pre-existing ideas of how genetic diversity is distributed geographically in some species. So some of my previous research looked at sampling some species across their ranges to see how that genetic variation is distributed. A good example of that is Asplenium flabellifolium. In this bottom right-hand corner map, I've got a map of how the sequences that are found, or DNA sequences, chloroplast sequences that are found in this species occur across Australia. So different colours on this map represent different DNA sequences. So for collecting in Victoria for this species, I'd want to collect like a population that has each one of these different sequences in it in order to try to represent that genetic diversity that might be underlying in that species well. So if you want to find out more information about that, that's the details of that publication there. So initially I'm going to have a target of around about 72 species in Victoria, and that initial target isn't all of the species in Victoria. You might remember that the total was over 100. Those species that I will target initially, they're species that I think that I will be able to find. There'll be species that we'll be able to get a reasonable number of spores from that we can manage and work with in terms of being able to manipulate them into containers and then be able to sow for germination trials. And there are also species that we will be able to germinate and then grow on later. Some species are quite tricky, so I haven't included that in that initial group of species. The initial group will also be both common and rare species. So we're not just focusing on rare species here. I'm going to focus on whatever species I could find and fit those criteria that I just described. So there are a few different ways that I could go about collecting the actual spores once we've found the populations in the wild. One is just to collect a voucher specimen to be pressed to get deposited in the herbarium. And how that's usually done is you collect your specimen and then press it between newspaper, cardboard, and 2 sheets of lattice wood. And then that dries the plant flat. And in the process, if it has spores on that frond, those spores will be shed onto the newspaper over a period of a week as the plant dries. So this picture in the bottom left here has Blacknum chambersii that have been pressed and dried, and you can see here where I've got the pointer. This is the spore print that's been left behind by this frond here. You can see that each penny of this frond, each segment, matches the impression of spores that have been left. So that was an example where I could just get it from the collection that we made for the herbarium specimen. And while that seems reasonably straightforward, that you just look for fronds that have spores on the undersurface, it's a little bit more trickier in practice than that, because a lot of the times the ferns will look as though they have spores, but they've actually already shed their spores, and you go to press the plant and there's no spores left behind on the newspaper. Sometimes you can get an idea of when the spores might be perfect for dropping onto newspaper. The color of the frond gives that away a little bit. Like sometimes the fronds tend to be a bit more of a black or a certain brown color when they're perfectly ripe. But that varies from species to species, so it is a little bit hard to tell in some cases, and there is a little bit of experience that you need to trial with particular species to find out when their perfect stage of maturity is. So when the spores do get dropped on the newspaper, then you can just collect them by folding the newspaper up and then flicking that newspaper until they all gather in the seam of all the fold of the newspaper. Another way that we've gone about obtaining spores for some of the fern species is through a process that's often referred to as orchiding. So in this case, entire plants are removed from the wild and they're grown on in a garden, which in this case is both the Melbourne and the Cranbourne gardens. And you grow those plants on until they produce fronds and then that produce spores. And then when those fronds have spores, then you could take those fronds and then repeat the process that I had in the previous slide. So remove the frond from the cultivated material and then press it between newspaper and sheets of paper, sheets of cardboard, and then wait for them to drop the spores onto that newspaper. There are a few advantages to doing this method. Like before, I mentioned that sometimes it's hard to predict when ferns will have their spores ready to drop onto the sheet. So if you've got the plant growing in your workplace next to you, then you don't have to keep traveling hundreds of kilometers to keep checking whether their spores are ready to be shed or not. You can just keep checking on a daily basis until they're at the perfect stage of maturity, and then you can collect the frond and then follow through the usual process of pressing it and waiting for it to drop in the dry condition. Another advantage is that you can continuously replenish your stocks that you've currently got. So that's a big advantage, for instance, if the storage protocols that you've been using aren't quite as good as they could be. So if I started storing spores in conditions that were showing up to be all non-viable after a while, then we've got a continual source there that we can keep sampling from to try other different storage conditions. It's also useful if you've got small species that will only shed a small number of spores in one shot. So for small species, we'll probably sample multiple times until we get a reasonable size collection that we can actually deal with better and have a little bit spare for doing trials and things with like that. And it's also a good source of new material for the gardens too. So if I've got lots of spores, I can germinate lots of plants that can then produce more spores and then produce plants with known provenance for the public collections in the gardens. So I'll just go through some of the current collections that we've got in the current setup. So some of the collections that we've kept track of where they were from. One of them is at Cranbourne Gardens here. This is a species called Adiantum formosum that was formerly throughout East Gippsland in the lowland areas along floodplains and areas like that. But with a lot of the clearing that's happened down there and also the recent bushfires, the range of that species has contracted considerably, and it might only now be found in just a handful of sites. So this is a really, I'm looking forward to getting spores from this species, and it currently is fertile. It's got spores that I keep visiting to see whether they're mature or not, but they still need another month or two to mature, and then I'll collect spores from that plant. This is a setup that we've got at the Melbourne Gardens. They're in the nursery. We've got this shade cloth set up and this framework over the top because I came in one day and the blackbirds had got stuck into them and started removing the plants from the pots. So we've put those over there, and that's seemed to have fixed that issue nicely. But when I remove this, that's what it looks like. It's just a whole heap of potted up ferns. So currently there's 40 species that we've got for orcharding, and 9 of those species have more than one population collected for it. So we've made a bit of progress for that so far. Here's some of the more eye candy like species that are represented among those collections. So we've got this species here, Pneumatoderus penigera. This is the lime ferns found in southwest Victoria or along the rivers down there, like the Glenelg River and Curtis River. I've got Asplenium hookerianum here. That's a species that's only found in 3 locations in the Victorian Alps. Asplenium ethiopicum, another species from the southwest of the state. Cystoderus tasmanica, a species from the high country. It lives along fast-flowing rivers and creeks. Plectrum deltoides, it's only known from Bryce Gorge, which was that place that I showed in the talk previously. That's this location for Asplenium hookerianum as well. And Pteris paleata, which is a species that I've recently described, split off from Pteris comans, which is now only found in the tropical Pacific. So all these species are threatened species, and they're all growing quite well in the Melbourne Gardens. So they're exciting prospects. So assuming that we actually get the spores off of the fronds to fall into the newspaper, the next step is to sift those spores through a 75 micrometer aperture sieve or mesh. So this is the sieve that I've been using. It's from CMT equipment. So if I just pour the spores and whatever other debris remains from the shed into the sieve here, and then sieve it through, I get this fine powder of spores coming through, and what's left behind is the debris, like the cases that the spores were contained within, the sporangia. So they get left behind because they're too big, they're larger than 75 micrometers, and other things like dirt and scales and hairs and things that are unnecessary, they get left behind in the sieve. And that's good because those larger structures could potentially cause contamination. So there's no use having them in the sample. So we want to isolate the spores from those before storage. We don't surface sterilize the spores. So in other things like seed storage, often they're surface sterilized to try to stop contamination. But previous studies have found that surface sterilizing in multiple different chemicals has reduced spore viability. So we're avoiding doing that. And you leave the spores to dry for around about a week at typical room conditions like room temperature and humidity. And that has in the past produced spore water content of around about 5 to 6%, which past studies have found to be about the optimal amount of water content to store spores at. So spores dry reasonably easily because there's not much to them. They've got a reasonably small volume, of course. So they dry out quickly. One of the interesting things that I notice about the spores is all the different colours that they produce en masse. So this is Dixonia spore here, and it's a really nice gold colour. You contrast that to Black Motzii here that's brown, and this species here is black. And there are other species that are green because they've got chlorophyll in the actual spore. This sample here I collected before we had the sieve, and you can see like you get all this other debris in amongst the spore mass. So there's bits of sporangia in there and possibly other bits like scales in there too. So you really do need a sieve if you want to get the spores by themselves. So once I got the spores by themselves, then I've been dividing the spores into small portions. So rather than storing them all as one giant mass, I've been allocating them into like aliquots in these parcels made of tin foil. And if you remember back to that slide with the microscope photo next to the ruler, I don't know how many spores that would be, but it would probably be in the thousands. So there's a generous number of spores just in that small parcel there. And if I collect or make a few parcels of those, then that should be ample amount for numerous trials and to germinate a whole stack of plants later on, because each spore has the potential in most species to produce a plant by itself. Theoretically, that's often not the case, but yeah, a lot of spores there for potentially a lot of plants. So once I've got this folded up tinfoil parcel here full of spores, I place it in an Eppendorf tube that's labelled with the sample, what it is, and then I put it in this cardboard box that's cryogenic grade protected. So I'll explain why that's important soon. And on the outside of the box, I've labelled exactly what's in there with pre-allocated sites for each species that I think I might be able to collect over time. And the positions are labelled on the inside of the box too. So how we've gone to think about how to long-term store these spores is we're going to put them in a minus 150 degrees Celsius electric freezer. and the company that we went with was Artico. This is the model here, Cryo 230. This is from Denmark. The reason why we went for an electric freezer is that instead of getting liquid nitrogen, which is often used for storing species that require really low temperatures, liquid nitrogen was not compatible to our area that we could keep them in the basement of the herbarium for health and safety reasons. So we went for an electric freezer instead. Previous studies that have looked at optimal storage temperatures for fern spores have found that over a broad range of different species, cryogenic temperatures, so those that are between -150 and around -196, have been the optimal for retaining spore viability over long-term periods of time compared to much warmer temperatures. That varies a little bit. So some species that are from dry conditions naturally, so some dry country rock ferns, can tolerate room temperatures for long periods of time and still be viable after that. But other species, like some species from wet habitats and also species that have chlorophyllous spores, are more sensitive to storage temperature, and they've been found to be retain more viability if stored at much lower temperatures, like what you see at cryogenic temperatures. So we've gone with the -150 degrees option so that we can potentially store a broad range of different species. So currently there's 7 species that we've got spore samples from banked in this freezer, and we've just started to put spores in this because we've only hooked this freezer up in the last in June actually, so it's only been running for about a month so far. So if I open the lid, this is what it looks like inside. It's got at the moment 2 vertical stacks of shelving, and that's where the boxes that I showed before slide into. I've put 2 separate shelves in for the moment. These are duplicates, so I've got one shelf here on the left that will have all of our long-term storage samples in there, and the shelf on the right has duplicates for trials and experiments and things like that. And the reason why I've separated them up so much is because previous studies have found that repeated cycles of freezing and thawing reduces the viability of spores. So I want to try to reduce that as much as possible, so that limiting the amount of time that these spores are out of the freezer by making sure that the boxes are labeled with the spots that they're supposed to go in, so that we know where exactly to put them and look for things rather than spending lots of time looking for them outside the freezer. And also having different allocations of the samples too, so that we don't have to take the entire sample out each time. I'm hoping we'll reduce that amount of freezing and thawing. Another important aspect of the spore bank is being able to germinate the spores. So that's the end goal eventually, that spores that are stored should be able to produce new plants that can then be used for reintroductions, etc. So I want to make sure that we're able to germinate them, and also that it's important to know that the spores that you are actually putting into the freezer are viable to start off with as well. So I've tried a couple of different methods so far for spore germination. One of the methods is just to do spread them on agar in a petri dish and then grow them on in a growth cabinet that has light and is kept at constant temperature. So this is an orthodox way that previous studies have germinated fern spores. It's good because it's highly controllable, but I've found that it's slow. So the germination rate's slow, and the gametophytes, which is the structures or the phase of the life cycle that the spore initially grows into has been really slow. So they haven't developed all that quickly, a lot slower than what I'd expect. Another method that I'm trialing is to grow spores on Dicksonia antarctica tree fern slabs, and then those are put in a closed terrarium that is kept constantly moist with really high humidity. So I've got misters in the terrarium, which is an exoterra terrarium, and that humidity is maintained at over 90%. And I've got lighting on it as well. So I've tried that as well, and I found that to be really successful so far, because I thought this is how a lot of ferns germinate in the wild. So I'll give it a shot in an artificial setting. And just within 2 months, I found from sowing the spores that I've got reasonable growth of gametophytes. So this picture in the bottom right, you see the individual gametophytes here that will produce the sperm and the egg to produce a new sporophyte, or like what most people consider a typical fern. And this has been successful over a range of different species too. So I think this might be the prominent way that I go about germinating more plants. So there are a few, this is all in its infancy so far. So we've only got a few different collections for spores so far, and we're going to keep going until I've got those initial collections all accounted for. But there are several extensions that we could think about in the future. One of those is looking at how to cultivate species that are traditionally been quite difficult to grow. So I've been experimenting a little bit with growing filmy ferns, which is the family Hymenophyllaceae, again in a terrarium with high humidity. And that's been successful over the last few years. I've been able to grow those with difficulty, mind you, but they are growing and surviving. But it's challenging in that they don't freely produce reproductive structures, and when they do, they're really small. So obtaining a reasonable amount of spores from some of these species is going to be really tricky. So whether that's going to be a reasonable prospect in the future or not, I don't know, but I'll keep having a look at it. And then there are other species that are difficult to grow, probably because they've got some sort of dependence on fungi for their germination and subsequent growth. So we know that in some members of the Schiziaceae and also the Glyciniaceae that's the case. So we might even have a look at germinating some with fungi and looking into what species of fungi might coexist with those plants and their roles and symbioses. So that's a few future research prospects in there. We'll also look at maybe storing fern spores from non-Victorian species. So at the moment, you remember how much of that cavity inside the freezer was left blank. Like I've got those 2 drawers that could contain all of Victoria's fern species. There's a lot of room left over. So if we make collections from non-Victorian species, then we could add those to the collection, to the spore bank. And also it would save other herbaria from having to purchase expensive freezers that require a lot of electricity. So it seems like a good thing to do. Look at other species as another potential prospect in the future. So bryophytes, for instance, that also reproduce by spores. They are difficult in the same ways that Hymenophyllaceae are, the filmy ferns, because they produce capsules that are really small and won't produce all that much spores all in one shot. And because of that, most studies that have looked at storing bryophytes long term have looked at storing their gametophyte tissues, which is the main conspicuous part of the bryophyte life cycle. But this is a little bit more complicated than in just storing fern spores, in that it requires culturing with various chemicals like abscisic acid and sucrose in order for them to be viable after subject to such low temperatures. Could also look at fungal tissue as well that might need low temperatures. Pollen, so cycad pollen is often stored at low temperatures. That might be a future prospect. And also non-orthodox seed as well. So most seeds are stored at temperatures that you might see usually in a fridge or at room temperatures, whereas some species that produce seeds might not be viable if stored at such temperatures. So trialing some of those species at really low temperatures might be a go as well. So that's what I've got today to talk about. I've got the following acknowledgements. So the Fern Spore Bank was funded by Dale Whip's second phase of bushfire funding. I'd like to thank Daniel Ballesteros from Kew for advice on spore collection and optimal storage conditions for fern spores. So he works on similar things over in Kew. And there's various contributors from Royal Botanic Gardens Victoria that have made this project possible. So Alistair organised the freezer. I've had lots of help out in the field collecting samples, so all those people there have helped with that. Lots of help out in the nursery and cultivating the plants. So David Robbins and Dermot for helping out in the nursery, and Misha, Tim and Scott for helping keeping tabs on some of those plants. And also thanks to Danny for helping me with some of the germination trials.
Thank you very much, Daniel. Looks like an enormous amount of work. You mentioned that there are something like 10 spore banks worldwide, I think. Are they using different methods to collect their spores, or are you all sort of working individually and working it out on a case-by-case basis?
I think it's largely case by case. I think from what I've seen, most people just collect the fronds and hope for the best and let them shed it onto the newspaper. And there's been a focus primarily on large species that's easy to do. So a lot of those spore banks overseas, they don't have a comprehensive representation of their fern flora. Instead, they have like a few species that they've been studying or that might be easy to gain spores from. So yeah, I think most of the time it's just been grabbing it from herbarium specimens or fresh, but just pressed and then dropped into the newspaper.
Yep. So arguably, this will be one of the more ambitious attempts at a fern spore bank.
Yeah. Yep.
Yeah, nice one. So if people want to write any questions in the chat, or if you want to turn on your microphones and cameras and ask Daniel any questions, we have about 10 minutes of question time if people want to chime in.
Chris, Tim here. Tim Emerson. Just a quick, simple question. That's good, Daniel. And I was wondering about the colour of the spores and whether you think that's going to tell you anything about their sort of longevity or the way they're stored. I was just curious whether, ones with chlorophyll in might not last as long or something like might need different temperatures. Does it help you at all?
So definitely with the chlorophyll aspect. So in general, spores that contain chlorophyll and end up green don't stay viable as long in general, and are more specific in what they require to stay viable. So they're very insistent on having those really low cryogenic temperatures to remain viable over many years. But as far as the other spores go that aren't green, I think the color gives an indication of how mature it is. So spores that aren't fully mature tend to be a lighter color and are potentially not all that great for storage. You definitely want to get things that are at peak maturity, but I don't think it really gives much of an indication on their best storage conditions. It's more environmental that has been the dictator of what to store in what conditions. So those drier country ferns that encompass multiple different families are better at withstanding storage at multiple different conditions than species from wet conditions.
Yeah. I was wondering whether, reflected some chemical in their wall that might help them last longer. It sounds like that's not the case, just age.
Yeah, not that I know of.
I might just ask a vaguely related question. You mentioned there are various techniques that reduce the viability of spores, but what sort of time frame are you looking at? How long will they last? And I imagine it will change depending on the genus and species, of course, but will you have a rolling testing phase to test whether things are becoming less viable over the years and then replenishing them at that stage? Or how will that work for the long-term storage?
Well, I suspect that we will have to start some trialing at a regular interval to see how their viability is changing over time. In previous study, because this is reasonably new stuff, like the research in this area of storage has probably only been really explored in the last decade or so. Most of these trials are over a period of up to 10 years. So we know that at least in some species, they remain viable for 10 years. And we suspect that they're going to last even longer than that. So that's what I would hope would happen. But with those benefits of having a source of new spores to replenish them with, then I think at least we've got the ability to keep adding to those collections in the freezer to make sure that they are always some viable spores in there throughout time.
Sounds like a perfect setup for a bunch of undergraduate projects as well.
Yeah, yep.
Dan, you've got your hand up.
Thanks, Chris. Daniel, I know with the wattle seeds, the tropical species don't really have any dormancy. Do you think will be similar with fern spores, that you'll get sort of lack of dormancy in tropical species compared to temperate ones?
I wouldn't imagine that there'd be all that much dormancy in any of the spores. They're such small structures that have no, like with seeds, they've got that outer tester or whatever to have to break, or some conditions for the embryo inside to start to activate. But in spores, it's just a single cell that I doubt there'd be all that much. As soon as they get the right conditions to germinate, so a moist substrate, I would imagine that they'd start germinating.
Yeah, interesting.
Austin, you have your hand up.
Yeah. Daniel, just a quick one. What levels of viability would you normally get at the stage that you're putting them into the freezer? You got some idea as to what range of viabilities you might be getting?
Yeah, so for some of those really difficult to cultivate species that I'm initially avoiding, probably really low, like 0 to maybe 10%. But for species that are easy to cultivate using normal methods, probably 80 to 90% germination success. So yeah, when I've sown them, they've produced a lawn of gametophytes. So that's high. That's really high.
Yeah. So quite a big range, and obviously, as time goes on, you'll probably work out some different techniques for the different species or different genera that would be the most advantageous.
Yeah.
Thanks.
Okay, I don't think we have any more questions in the chat room. Just give it another minute for people to interject. The final questions? All right, thank you very much, Daniel. Most appreciated.
I just saw a question in the chat that says any trends with classification, I think.
It's not coming up, but mine. Apologies to who wrote that.
I don't know if I've looked at enough to be able to make a really good call on that, but I don't think so. I think it's mostly germination requirements and storage temperatures is more correlated with what environment the plants were from rather than what they're related to. But there could be minor things, minor effects due to classification, but at the moment it looks more environmental than anything.
Now that my questions, the chat has come up for me as well. I see your question, Casey. Sorry about that. Casey asks, are there any spore-borne diseases like house pathogens that are dormant in seeds? And if so, how would you screen for healthy spores?
Not that I know of. In hybrids that are infertile, the spores will be malformed, so they won't have the usual shape and they often look like a grain of dirt. But that's not really a disease. That's just because they don't form properly. Yeah, so far all that I've seen, they all look somewhat similar when they're fertile. So no, I don't know about anything like that.
Okay. All right. Now, thank you very much, Daniel. I don't think there are any further questions. So that wraps up our 6th seminar. Our next seminar in August will be presented by Tara Hopley, who's a postdoctoral researcher at the Royal Botanic Gardens Victoria, and she'll be discussing some of the work she's been doing on a large-scale genetics project following the relatively recent bushfires. So thank you very much all for coming once again, and hopefully I'll see you next month for seminar number 7.